Hi everyone, Yusuke Marikawa here, and this is a study guide for the upcoming quiz 6, which is on April 1st. Okay, so usual 10 MCQs, which covers the 5 lectures that I gave, so listed here. So those 5 lectures only, okay? And in this video, I'll specifically focus on what aspect you should study very hard, and some things you need to memorize I'll point out. So you can dedicate your studies on those points particularly. And so that will help you to score pretty good in the quiz 6. Okay. So the first lecture, that's amino acid catabolism here. So let's see. Okay, so this is the protein digestions, the dietary protein. So you need to know those 3 organs, right? Stomach, pancreas, and small intestine. Those are the ones that produce those proteolytic enzymes to digest those foods. And what you need to associate is the organ and what enzymes are produced or secreted. Stomach for pepsin from stomach, trypsin from pancreas, don't worry about the others, and aminopeptidase, intestine, right? And also, kind of relation to that small intestine part, you need to know this disease, what this is. Celiac disease, which is an autoimmune disease, and your antibody reacting to this gluten in your food. And that causes inflammation followed by the atrophy of intestinal cells. Yeah. So this disease, you need to be aware. I mean, what is this disease is about. Okay. And also, you need to know how the turnover of endogenous proteins occurs. This is one mechanism that I emphasize. You need to know the term, ubiquitin and the proteasome. Basically, those unneeded proteins are tagged with this little protein called the ubiquitin. And that tagged proteins recognized by this giant protein destruction machinery, this proteasome, okay? Ubiquitin proteasome system or turnover of endogenous proteins. And you need to know this process of removing the amino group from amino acid. Basically, that's the first step to use this amino acid for energy production purpose. So the amino group removal. And that involves the 2 processes, the transamination and oxidative deamination. You need to know those terms, okay? Transamination uses this aminotransferase or transaminase enzyme, okay, that's passing this amino group to alpha ketoglutarate. And then second part, oxidative deamination, okay, oxidative deamination by glutamate dehydrogenase. And that's the one that will remove ammonia from glutamate, okay? And for the transaminase, the one I just mentioned here, that requires this cofactor, PLP. So you need to know about this. This is an essential cofactor to carry out this transamination reaction. You need to know what this is derived from, vitamin B6. Okay, particularly this, you need to know pyridoxine, but vitamin B6 give rise to PLP attached to this aminotransferase to help this enzyme to carry out this transamination reaction. And so some of the this conversion, okay, this after transaminations, you end up with some compound. So you need to know the relationship between this amino acid and the resulting this compound. So alanine to pyruvate, you need to know, okay, alanine losing this NH2, it becomes pyruvate. An enzyme for that is alanine aminotransferase, or ALT. You need to know the full name as well as this abbreviation, which is good. And good for your, you come across with this ALT things in the future. So it's good to know. Alanine to pyruvate after losing this amino group in this transamination reaction. I emphasize this part, but you don't need to memorize all those amino acids that will give rise to pyruvate. But you need to know pyruvate is an amazing thing, okay, because it can do lots of things. Particularly, you need to remember this pyruvate can be used to make glucose through this gluconeogenesis, right? Pyruvate can be converted to glucose through gluconeogenesis. That's why it's important to know that certain amino acid can become pyruvate because those amino acids can become glucose through this process, okay? And also aspartate to oxaloacetate, and a similar relationship. Alanine to pyruvate, aspartate to oxaloacetate. Enzyme is a little different for that. Aspartate aminotransferase, AST. Yeah, remember. And also this enzyme that dedicated for the metabolism of the branched chain amino acid. So you need to know those three amino acids that are categorized as branched chain amino acid, valine, leucine, isoleucine. Those three, you need to know those three amino acid names. Okay, those are all branched chain amino acid, BCAA. And there are specific enzymes involved in the metabolism of those branched chain amino acid, which is this BCKDH. Just be able to recognize this full name as well. But the BCKDH, that's enough. And important, the reason is that why it's important is that there's a human disease that caused by the mutations or deficiency in the BCKDH, that very enzyme that I just emphasized, which is maple syrup urine disease. Okay. And this is basically that the person is lacking this BCKDH, unable to process that branched chain amino acid. So ends up accumulating those BCAA. And some amino acid is the cause of that maple syrup-like smell. And some amino acid causes neural damage. You don't need to know that detail, which amino acid, but just make sure that you can relate those disease name and the cause of it and what's the outcome of that enzyme deficiency. Basically, an accumulation of those BCAA, the three amino acids that I just mentioned. Yeah. And you should be able to identify accumulation of which amino acid. Yeah, plus any of those three branched chain amino acids. All right. Okay. So moving on to the next lecture. So let's see. So the focus is the production of urea. So this entire slide is actually very important. Yeah. And for example, this production of urea happens in the liver. Okay, liver is the only organ that produces this urea, which is the way we mammals dispose this nitrogen waste like ammonia. This is a very toxic compound that results from the amino acid metabolism. So we take care of it by changing into the safe compound, urea. Okay. So to make this urea, the safe urea, the several molecules involved, ammonia, of course, CO2, and aspartate. So the second amino group is from this aspartate. Okay. And also know that it consumes ATP, yeah, energy consuming reactions. Okay. And the first step of this changing ammonia into urea is this process, okay? Production of carbamoyl phosphate, okay? Carbamoyl phosphate. And then CO2, NH3, ATP, and of course, water is involved as well. Yeah, so that will make this carbamoyl phosphate. Enzyme name is important. It's a straightforward. Yeah, carbamoyl phosphate synthetase 1, okay, CPS1. For this quiz or exam, that's enough. But later, the final exam, there is another enzyme called the CPS2 coming up. So it's good to be aware or make sure that you connect the CPS1 to urea cycle. Okay, CPS2 is for something else, which is going to be important later on. But for this quiz, just carbamoyl phosphate synthetase. Yeah, if you can recognize that, that should be enough. And this enzyme, this step is the rate limiting step. So it's going to be some regulation happens. I will emphasize that later on. This is the very important first step or rate limiting step of urea production, which happens in mitochondria. mitochondria. Okay. So that's happening in the mitochondria, making this carbomoyl phosphate. Then the following the cycle, this is the key part of this urea cycles, eventually releasing this urea. So what you definitely need to know in this slide, it would be nice to know everything here. It's not too complicated, but particularly you can definitely need to know it's this ornithine. Ornithine is the one that received this first amino group from the carbomoyl phosphate. And then the final step is from this arginine to ornithine, back to ornithine. So this process will release that very urea. Yeah? Arginine carrying 2 amino group, release this urea and go back to this ornithines. Yeah? Arginine to ornithine, that process will release this urea. Okay? So this is just to know about the fate of this aspartate, which is involved in the urea cycle. The second amino group of the urea is from this aspartate, right? So you can kind of associate that after donating this amino group, aspartate will become fumarate in the context of urea cycles. Okay. But transamination, a few minutes ago, I emphasized that aspartate will become oxaloacetate. Yeah, that's transamination. Yeah, giving away the amino group, but giving to the somebody else. Yeah, so that process is different. Yeah, so the outcome is different as well. So make sure you associate in the urea cycle, aspartate will become fumarate, and in transamination, aspartate will become oxaloacetate. Okay. And so this regulation I mentioned, the first step, rate limiting step of the urea production, CPS1, and that's regulated or activated by this N-acetylglutamate. It's key regulator. You need this N-acetylglutamate to activate this CPS1 to start this urea cycle. So definitely you need this compound, N-acetylglutamate, right? Now, so this slide is describing how nitrogen waste, that are produced by various organs and tissues like muscles need lots of ATP. So lots of amino acid metabolism releasing ammonia, but it need to be delivered to the liver to make urea, but you don't want to dump those toxic ammonia into the blood. So we'll deliver this nitrogen waste safely to the liver through this glutamine and alanine. Okay. Just remember those 2 amino acids name as a way to deliver nitrogen waste safely to the liver. Okay. And a little more details are described here, but you don't need to know that much detail. It's kind of this straightforward, but glutamine serving as a sort of transporter of nitrogen waste, in a safe way, and alanine same way. Yeah, that's the very important aspect. Okay, it's good to appreciate this cycle thing or mechanism, but need to identify those what amino acids are serving as a safe way to transport this nitrogen waste. Okay. So hyperammonia, so that's a disease condition, yeah, and caused by the high ammonia. Yeah, that's the name of the condition caused by the high ammonia, right? It's bad for your brain. And there are 2 kind of types of hyperammonemia. Okay, acquired hyperammonemia, congenital hyperammonemia. So be able to distinguish between 2. So acquire, usually through, during your lifetime, you damage your liver. And since the liver is the only organ to process ammonia into urea, so the defects in the liver will lead to acquired hyperammonemia. So anything that will damage this liver, like a viral infection or too much alcohol consumption, damaged liver leads to acquired hyperammonemia. And congenital, basically, you're born with it. So the gene mutations, the urea cycle disorder. So if you are born with a mutation in the gene that's essential for urea cycle, leads to congenital hyperammonemia. Okay. And there are 6 enzymes that are involved in this urea cycle, even though all of them are important, but what's most relevant for this quiz exam is, of course, CPS1 I emphasized earlier on, but here, OTC, ornithine transcarbamylase. Ornithine transcarbamylase. This is the most abundant or most common urea cycle disorder in human. So it's worth remembering it. Okay. Inherited deficiency in urea cycle enzyme like OTC or CPS1. Okay. Ornithine transcarbamylase. The one that's transferring this ammonia or amino group from the carbamoyl phosphate onto this ornithine. Yeah. And the medication, yeah, there are several medication, but the one I described in the lecture, yeah, for the hyperammonemia, that's phenylbutyrate, okay, phenylbutyrate. Know the mechanism of action of this drug, okay, how this drug will eliminate this nitrogen waste from your body so that the ammonia level is going to go down. So basically, this metabolite of the phenylbutyrate in your body will react with glutamine. Okay, glutamine. That's the one that's transporting this nitrogen waste. So react with it, get combined with it. Yeah, so that's going to be easily excreted in the urine. So the kidney will pick this up and dump in urine. So that's how you discard this glutamine. As a result, you will lower nitrogen waste level, therefore lower ammonia. All right. Okay, moving on to the next one, amino acid synthesis. Okay, you need to know the names, or at least you should be able to identify which amino acid is non-essential versus essential. Yeah, there's a list of multiple choices of amino acid. You have to pick, if you ask which is essential, which is non-essential, you should be able to pick right amino acid. Okay, memorize them and any way you can. I mean, probably I think this is gonna easiest way. Anything starting with A, anything starting with a G, those are non-essential. And P, S, Y, C, psych. Yeah, if you're familiar with a single letter abbreviation, psych will basically stand for all those amino acids, and they are non-essential. So anything else, anything other than those AAA, GGG, psych are essential amino acids. Okay, so they are essential because they cannot be made or synthesized in human body. That's why it's essential. So some of the this synthesis process you need to know, which I'm going to emphasize or point out. Glutamate can be synthesized from alpha-ketoglutarate. Okay, glutamate from alpha-ketoglutarate. Through the process that are kind of opposite of the oxidative deamination that I emphasized early on by the same enzyme, glutamate dehydrogenase. Glutamate dehydrogenase. One important thing here is that it will use NADPH as a power source to connect this ammonia, free ammonia, to this alpha-ketoglutarate. That requires some power. Yeah. So NADPH is the one that's used to bring in this ammonia to make this glutamate. Okay. So the some of the amino acid made from those intermediate of the glycolysis or TCA cycle, and you need to know some of those key ones. Alanine is made for pyruvate, aspartate made from oxaloacetate. That's the same relationship when you just the one you just went through in the transamination reaction. So it's easy to remember, right? Alanine to pyruvate. Those are interchangeable, aspartate to oxaloacetate. Also, how serine is made, you need to know. It comes from the glycolysis intermediate or 3-phosphoglycerate. Okay, phosphoglycerate becomes serine, yeah, through this process. But what you need to know the start point and the end point, and also this is a good example of the sort of self-regulation of the process. Yeah? So the end product of this reactions, which is serine, inhibits phosphoglycerate dehydrogenase. Okay, phosphoglycerate dehydrogenase. That's the start point of the conversion of the phosphoglycerate into serine. So if you have lots of serine, you don't have to change this in precious phosphoglycerate for serine production purpose anymore. So that'll regulate by feedback, negative feedback like this. Okay, so this process, glutamate to glutamine, this is done by glutamine synthetase, and this is the enzyme that will bring in this ammonia. You can catch and bring in this ammonia. It's gonna sort of important reactions, yeah. Glutamate dehydrogenase I mentioned earlier on, that's also capturing ammonia using NADPH. But in this case, glutamine synthetase will bring in ammonia, had a lot of energy here, and changing glutamate into glutamine. So the glutamine ends up with extra amino group here. Okay, using ammonia to add one more amino group, glutamine synthetase. Okay, and let's see. Okay, so the glycine can be made from serine. Okay, glycine made from serine by receiving by giving away. Okay, giving away is one carbon to tetrahydrofolate. So one carbon is transferred. One carbon of the serine transferred to tetrahydrofolate. It ends up glycine. Okay. So this process, this statement here is probably sufficient. You can see that this portion is transferred onto this THF. So I'll emphasize THF later on again, but this is one of those THF involving reactions, making of glycine from serine. And then tyrosine can be made from phenylalanine, okay, phenylalanine, which is the essential amino acid, but the phenylalanine to tyrosine, the enzyme is phenylalanine hydroxylase, hydroxylating phenylalanine to make tyrosine. Okay. Know the enzyme name and also the cofactor, okay, cofactor, biopterin required for this reaction. And this is related to this disease, phenylketonuria, okay, phenylketonuria or PKU. Okay. This is a disease, like a genetic disease, or could be some other deficiency problem, like defective phenylalanine hydroxylase that I just talked about, mutated, defective, that leads to phenylketonuria, or the person has, is deficient in this biopterin. Okay, so biopterin deficiency, can't, so that you cannot form this conversion of phenylalanine to tyrosine because this enzyme requires this biopterin. Yeah. So this is not a good condition because accumulation of this phenylalanine will eventually damage your central nervous system. Okay? Phenylketonuria caused by either phenylalanine hydroxylase deficiency or biopterin, the important cofactor deficiency. Okay. And one more thing you need to know is that, some essential amino acid, humans or the animals cannot make, but plants can make, like a tryptophan, phenylalanine. Those are, for us, essential amino acid we cannot make, but the plants can make. But they have to go through this very complicated reaction, which we abandoned long time ago in evolutions. So you need to just know that, at least recognize this EPSPS, one of the key enzyme in the production of this tryptophan, phenylalanine, this amino acid in plants, because this enzyme is the target of this Roundup, this weed killer. More specifically, these chemicals in this Roundup formula, it's called a glyphosate, and that inhibits this EPSPS, so that plants cannot synthesize those, phenylalanine, tryptophan, those important amino acids for them, so that they're gonna die. So that's the mechanism, So you should be able to associate this chemical in that weed killer and that inhibits this EPSPS, which is essential to make those amino acid phenylalanine tryptophan, of course, tyrosine as well, in parts, right? Okay. Next is this some amino acid derived biomolecules, part one. Here you need to know that this neurotransmitters production, how we produce those neurotransmitter from existing amino acid. So some of these key things that you need to know is that which neurotransmitter comes from which amino acid, the relationship that you need to know. Like serotonin is from tryptophan. Serotonin is from tryptophan. And also the key enzymes involved aromatic L-amino acid decarboxylase. Yeah? Aromatic L-amino acid, some amino acids that contain ring structure, aromatic ring structure, those aromatic L-amino acid decarboxylase, removing carboxyl group. Yeah? So the tryptophan choose serotonin by this decarboxylase. And also this biological relevance of the serotonin event. Serotonin is important for controlling mood, appetite, sleep, but the mood aspect is linked to this medication called SSRI. There are various SSRI group of medications that many people are using, like a Prozac. Yeah, this is one of this SSRI, selective serotonin reuptake inhibitor, but you just recognize as SSRI. And which is the inhibitor of the serotonin transporter, because serotonin transporter is the one that retrieving or eliminating this serotonin from the synapse, so that the serotonin is not going to hang around too long. So by inhibiting this transporter, you can keep the serotonin level high at the synapse. So that's how you maintain the active serotonin signal for a while in this patient to help maintain mood. So that's how SSRI works. So you need to know those yellow highlighted or box things at least. And the relationship between this two, tyrosine, dopamine. So the dopamine neurotransmitter is made from tyrosine. For this one, you also need to know the intermediate name, L-dopa. Forget about this long actual name, but the abbreviation at least, L-dopa. And the same enzyme, this aromatic L-amino acid, this ring structure aromatic group, and the carboxylase is also carrying out this conversion here. Okay. And dopamine is an important neurotransmitter that are linked to that cause of this Parkinson's disease that causing shaky hands or uncontrolled movement, so on. So this is caused by the low level of dopamine because the person lost or degenerated these neurons that produce dopamine in the brain, okay, caused by the low level of dopamine. Okay, and I point out L-dopa because it's medications, but that's the kind of extent of what you need. At least associate dopamine to Parkinson's disease. Okay, probably I'm not gonna ask that detail about this. This is kind of cool stuff, but probably not gonna be on a quiz. Okay, adrenaline or epinephrine. There's also the neurotransmitter and norepinephrine as well, right? Kind of easy to remember was epinephrine or adrenaline, same thing. It's from this dopamine. Okay, that means ultimately it's from tyrosine, tyrosine to dopamine, dopamine to epinephrine. Also, the final neurotransmitter is a histamine. Histamine is from histidine by this histidine decarboxylase. Yeah, kind of easy to remember. Histidine, right? And probably this histamine as involved in allergic response. So there's a medication to treat that allergy, antihistamine. But the histamine is also a neurotransmitter that controls the wakefulness to keep you awake in the brain. So that's why some of the antihistamine, when it enters the brain, it causes the drowsiness. So that's why it goes into the brain to inhibit this histamine signal in the brain, cause the drowsiness. Know this kind of stuff. This kind of summarizes the relationship, but I already emphasized that. Now, glutathione, know that it's an endogenous antioxidant that will neutralize the reactive oxygen species. That's very reactive. If you have too much, it's going to be dangerous, those things in your body. Know that it's made of glutamate, cysteine, glycine. Yeah, know that. And also know that it has the thiol group or SH group. Okay, that's the kind of key elements of the glutathione's actions. Okay. So the way this glutathione is neutralizing reactive oxygen species like H2O2, hydrogen peroxide, that's one of the reactive oxygen species, is through this glutathione peroxidase. So, this reduced form, the glutathione is reacting with H2O2 and becomes this, and the H2O2 becomes the water, safe water. So that's how glutathione neutralizes ROS. And this enzyme, you need to know, glutathione peroxidase. And that's the one neutralizing ROS with glutathione. So here, the cofactor here, which is actually a part of this specific amino acid selenocysteine, but selenium, which is the type of metal, yeah, it is a part of this glutathione peroxidase. And that is essential for this enzyme to carry out this reaction. So this is you can call it cofactor, probably easier to remember. Yeah, selenium serving as an essential cofactor for glutathione peroxidase. Okay. And once the glutathione is used, you want to restore this glutathione into this reduced form, which is kind of active form, through this glutathione reductase, reducing back to this active form. And here, to reduce to the active form, you need some power, which is NADPH, which is produced through pentose phosphate pathway. Pentose phosphate pathway produces NADPH, which helps glutathione reductase to rejuvenate glutathione. Okay. Like nitric oxide, there's a simple kind of signaling molecules, NO, and which is the biological function causes dilation of blood vessels, vasodilator. Yeah, kind of expand or relax the blood vessels so that the more blood flow, vasodilations. And know that nitric oxide is made from arginine. So tiny molecules, nitric oxide, but it actually come from this amino acid arginine by this nitric oxide synthase. But arginine is the source of this nitric oxide. And the action of nitric oxide, you need to know that it's basically to increase cyclic GMP, by activating this guanylyl cyclase. So this enzyme will change GTP into cyclic GMP, which leads to the following cascade that eventually leads to vasodilations. NO activate guanylyl cyclase to increase the level of cyclic GMP, okay, to cause vasodilation. And some medication that, for the heart pain, and which is caused by the narrow blood vessel. So nitroglycerin basically will release this nitric oxide. With this breakdown of this nitroglycerin will release nitric oxide, which will relax this blood vessel to increase the blood flow to help these patients. Also, some clinical correlation is that this male erectile dysfunction medication, Viagra or sildenafil, is actual chemical name, and more brand name is Viagra. But anyway, the action ultimately is to increase cyclic GMP, right? So that you want to increase the blood flow into the penis and to sustain the erected state. So to increase cyclic GMP, it's basically inhibiting this enzyme, PDE, phosphodiesterase. So you need to know this enzyme name here, phosphodiesterase, because this phosphodiesterase is destroying this cyclic GMP. Okay, so the Viagra inhibiting that phosphodiesterase, you are keeping that cyclic GMP level high, vasodilation. Got it? All right. So the last one. Okay. Here. So just I talked about this homocysteine. That was the sort of focus here, homocysteine. Okay, so there are three kind of amino acid. All of them are contain sulfur. Okay, SSS. Cysteine, methionine, you're familiar with. That's the kind of regular amino acid that's going to be a part of the proteins. Homocysteine that appears in our body, but it's not a part of the protein. So non-proteinogenic amino acid, homocysteine. But it has kind of important role, appears in the various reactions. So that was the focus. So be able to recognize these amino acids are sulfur-containing amino acid. All of them, all those three containing sulfur. And also know the name of these chemicals, the S-adenosylmethionine or SAM, okay, as the methyl group donor. Okay, SAM as methyl or CH3 group donor. It donates to the various molecules to change the activities. Yeah. And about oh yeah, so this, after SAM giving away, eventually become homocysteine. Now I talked about this, how this homocysteine are sort of reused or converted back to the methionine. Precious methionine, you're restoring, rejuvenating from homocysteine. There are two pathways. One is to receive this methyl group from tetrahydrofolate. So this is the reaction. So what you need to know is that homocysteine to methionine requires this the enzyme, this methionine synthase requires two cofactors. One is folic acid or vitamin B9. The other one is cobalamin vitamin B12. So the methionine synthase requires these two cofactors to give this methyl group to homocysteine to restore this methionine. And no, this tetrahydrofolate I mentioned earlier on, yeah, that's derived from folic acid, vitamin B9 in your food, and that serves as a one carbon donor or acceptors. There are various forms of the carbons here, one carbon here, but all basically this tetrahydrofolate is receiving or giving away this one carbon. That's the major role of tetrahydrofolate in the various reactions. And restoring methionine from homocysteine. Number two, this one is receiving methyl group from trimethylglycine. Yeah. And what you need to know here is that this trimethylglycine comes from choline in your diet. Yeah, choline. Okay. So choline, yeah. So choline is used for those methionine rejuvenations and so on. Here, what's important is choline is serving as a precursor for the very important neurotransmitter, acetylcholine. Yeah. By receiving this acetyl group from acetyl-CoA. So the choline plus acetyl-CoA, you can make this acetylcholine. Yeah. Important neurotransmitter. And this neurotransmitter, acetylcholine, acts through two kinds of receptors. Just be able to recognize the names of the receptors. Okay, the actual mechanism of action is okay. Kind of cool things to know, but right now it's okay. Okay, nicotinic receptor and muscarinic receptor. So just be able to recognize those terms as acetylcholine receptors. Okay. So the production and the breakdown of this acetylcholine, this is happening at the synapse here. So the one making acetylcholine is choline acetyltransferase. And the ones that destroying or inactivating this acetylcholine is acetylcholine esterase. Okay, acetylcholine esterase. Yeah. So be able to recognize those enzymes and what they do. Yeah, making acetylcholine, destroying this acetylcholine. Okay. So this acetylcholine esterase, that's the focus now. This enzyme can be the target of certain medications. For example, this Alzheimer's disease medication. One example is donepezil. This is FDA-approved medication for Alzheimer's disease. And basically, it's inhibiting this acetylcholine, which prevents from the destruction. So as a result, this drug will lead to this increase in the acetylcholine level. So that basically helps brain to act better with more acetylcholine, to sort of alleviating the symptom, that's dementia symptom of Alzheimer's, right? But if it's too strong, this inhibition is going to serve as kind of poison, like a nerve agents. The examples are some pesticides, some nerve agents, like chemical weapons, sarins. They are collectively known as organophosphate because of these structures. Okay, so be able to recognize organophosphate as inhibitors of very strong inhibitors of acetylcholine estrase. Okay, can be found in the certain pesticide and the chemical weapons. And need to know there are some antidote for this organophosphate poisoning. Yeah, so I talked about those two chemicals in this antidote, but this is atropine. You need to know which inhibit this muscarinic receptor, this whole thing. Okay, muscarinic receptor, acetylcholine receptors is the target of atropine. So the too much acetylcholine is the causing this toxic action. So by preventing that action of acetylcholine by inhibiting this receptor, you are alleviating this toxic excessive level of acetylcholine signaling. Got it? And that's it. Okay, so those 5 lectures I just went through, those things I emphasize here are very, very critical for this upcoming quiz 6. And tell you the truth, the ones I just emphasized, it's going to be super, super relevant for the upcoming exam 3 as well. Yeah. So study hard for the quiz 6, but even after that, continue studying using this guide for the exam 3 as well. You know, super, super important. Okay. All right. So that's it. But if you have any questions, contact me. This is my email address as usual, right? Study hard. Yeah, I promise you get good score if you did more study based on following this my guide, the higher your score will be. All right. That's it. Take care.